What do we think is actually causing aging?
Can we stop aging? Can we reverse aging? Maybe the most important question in biology, all of the interventions that extend lifespan reduce chronic inflammation.
But if we fundamentally want to get to a point where maximal human lifespan is changed and health span is fundamentally altered, we have to bend the slope of that line.
Yeah.
What is the probability in your mind that rapamycin is doing that?
We're doing a study like this in Singapore on humans.
Okay.
It's a 6-month intervention with rapamycin.
I'd like to just quit my job and do nothing other than these experiments. Did you guys do a study on urolithin A as well?
We haven't published it yet, but I am happy to talk a little bit about it. We don't have human data, but the mouse data is really good on urolithin A. I think a lot of these supplements are impacting exercise, and so it's kind of a win-win.
There's so many ways to just help a person find another 5 years of life and dramatically improve health span. Do you think that immortality is impossible? Hey everyone, welcome to the Drive Podcast. I'm your host, Peter Attia. Brian, thank you so much for being here. You might actually hold the record for longest journey taken to come to this podcast. In fact, I don't know if anybody could travel a greater distance than from Singapore to come out here. So thank you very much.
Oh, it's my pleasure to be here. And I don't think you can get further from here to Singapore.
So let's tell folks a little bit about how you wound up in Singapore. I guess just I'll speed things through a bit, but by way of background, obviously, you used to run an institute called the Buck Institute. Mm-hmm. Tell folks a little bit about what the Buck is and what you did there.
Yeah, it's a fascinating institute because it was really the first institute solely devoted to understanding aging and longevity, and it started in the around 2000 with some money that was donated by a woman who died in Marin County, north of San Francisco, and I was the second CEO there in 2010. There were about 20 faculty at the time, all devoted to either aging or aspects of aging. Very basic science. And as you can imagine, in the 2000s and around 2010, that was a significant component of the aging research field. It was still a very small field. And so the goal was really to help that institute grow. And it was tough times in the 2010s because the funding levels were low. And it was right before the real interest in aging and longevity happened around 2017, 2018. So, we were really struggling to keep the doors open. And I think the Buck's doing a lot better now, and as well as the rest of the aging field.
At the time that you were there, how much of the funding came from NIH and how much came from either donations or industry?
Yeah, it was very heavily oriented to NIH, and our goal was to get more industry funding. We started 7 companies when I was there, some of which are still hanging around, and also really tried to ramp up the philanthropy. But, philanthropy for aging wasn't really happening until around 2017, 2018, when people started really getting the idea that you could slow aging and prevent all these diseases and stay healthy and functional. And so that revolution happened around that time.
The faculty there it's quite the star-studded cast, right? I mean, Eric Burden's there, Judith Campisi was there until she passed away.
Yeah, that's unfortunate.
So I mean, you really had a collection of people doing great work, right?
Yeah, well, Eric came after I left. He took over as the next CEO. But yeah, Judy was there, Henry Jasper. He's gone on to Genentech. But Gordon Lithgow and a bunch of other people working on aging. So it was a good group of people for sure.
What do you think led to this interest that you've alluded to in 2017, 2018 in this idea of, you describe it as you see fit, but something happened in 2017, 2018 that's brought a lot more interest into the field, however one describes it?
I think you just reach an inflection point. It's really hard to know what triggers it. Calico started a few years before that, were Google's alphabet companies that was focused on longevity. They've not been very open about what they're doing, but it triggered a lot of publicity for the longevity field. It got Silicon Valley interested. And I think that Matt and I used this slide that you have aging, pointing to all these different diseases. We started using that slide around 2005, and we were making the point around healthspan shortly afterwards. And, really this idea of preventing disease and keeping people healthy, interacting earlier. And I don't know, I'm so sick of that slide, I can't look at it anymore. But it's finally, and we weren't the only ones doing it, but I think there were a few of us doing it. And it really finally, I think, helped trigger a movement, hopefully. So,
do you think the field would have, and these are sort of dumb questions as I realize as I'm asking it, but do you think the field would have accelerated sooner had it not been for some notable setbacks? For example, I don't remember exactly when GSK bought resveratrol, but I believe it was like around 2006, 2007.
Yeah.
It was clear to me, I think, by about 2010 that was not going to work. And I think it was probably clear to GSK around that time as well.
I think.
Yeah, maybe sooner. So do you think that type of hype with nothing to show for it was a kind of a negative force in that equation? And maybe this inflection point could have happened sooner, i.e. during your earlier tenure there, could it have been easier to have raised funds if there had been less of those examples?
I think it's hard to know. I mean, it's unfortunate what happened. I mean, in one way, the investors made money off of that deal. And so, that in that sense, it was a success, but what was developed was not going to go anywhere. And that's unfortunate. I think that it probably slowed things down a little bit. Yeah. And I think because there's always this doubt about whether you can slow the aging process. And so when you have a major, effort that's triggered around trying to do that, even though they ended up focusing on disease, and we can talk about the struggles of longevity biotech companies in that way. But when something like that, fails, it probably does slow down other investor interest.
Today, you're in Singapore. Tell me what you're doing there.
So, I kind of have one foot in academics and one foot in the private sector these days. On the academic side, we're really focused on targeting aging. And, and that comes back to what I alluded to with the biotech companies a minute ago. A lot of them are targeting aging pathways, but to raise money and get their drugs tested, they have to turn to some disease indication, which is understandable. And companies I'm involved with do that too. But that's not what we really want to do. What we really want to do is slow the aging process and keep you from getting sick. And so in an academic setting, we can test that clinically. So we basically have a whole range of animal models, a pipeline from yeast, worms, flies, killifish, mice, and humans. And
billion years of evolution there.
Yeah. We bring interventions in and at the right place, validate them, really believing the idea that if it works across different model organisms, it's more likely to work in humans. And then we design human clinical intervention studies to validate that they're targeting the aging process. I don't think anybody knows exactly how to do that yet, including us, but we're doing our best and learning as we go.
How is it funded, the institute you're at?
Yeah, mostly through the university and the government in Singapore, but we also have some philanthropy, and we do contract-sponsored research to test interventions from companies as well sometimes.
How many PIs are there?
So I run a program that has about 35 PIs in it.
Wow.
But a lot of them are doing other things. They're not all focused on that one concept I just told you. They have their projects around Alzheimer's disease, or we just have this guy, Michael Chi, who's working on sleep and aging, which is so understudied, and so it's kind of like an academic department. People have their own projects they're focusing on.
Is there any department at a U.S. university that brings together as many people that are focused on specifics of aging this way?
Yeah, I don't think so. Although I would say it depends on how you define aging. You know?
Yeah. If you branch it out to cancer, and things like that,
then definitely. Yeah. But I'm not sure if it's this focused on actual aging process.
So let's kind of start with a question that I think we'll end up coming back to because it's so fundamental. I enjoy, I enjoy going down the rabbit hole of fundamental questions in physics.
Sounds good.
But we're not going to do that. But I think the fundamental questions in biology, I think some of them center around aging. And so what do we think is actually causing aging?
Well, okay, so I'm going to force you into physics since you asked that question.
All right, very well.
Because we just had the first ever international conference on gerophysics in Singapore. And I was one of the organizers, and the reason I got behind it is the very question you just asked. We've been debating what aging is for the longest time. And, I think we would argue for 2 hours in some conference room somewhere in the world. And at the end of it, we would come up with the definition, shit happens and then you die. It's really just, it's frustrating. I don't even want to talk about it now. And, Vadim Gladyshev has been on like a evangelical rant about how do we define aging? We don't know how to define it. He asks that question at every conference. And I think it's a fair question. And we all throw our hands up in the air. And so the idea was we have a lot of data now, a lot of human data. And aging researchers are beginning to try to model that data. But they're not modelers. I think most aging biologists, or at least myself, if I have a skill, it's intuition. It's not writing equations and code, but the physics people, the theoretical physicists especially, they know how to model things, and they model things based on physical principles that are proven, right? And so we've been trying to bring these groups together. And because I believe maybe the only answer to your question is that we have to write it in equations. And so early days on that, but I'm excited about where that's going.
And do you think that these will be explainable through equations, or do you think that this exceeds our level of intelligence to understand, and it's really going to be up to a black box that contains a neural network to understand this? And maybe we take a step back, actually. So for the listeners, they've heard us on this podcast talk about quote unquote hallmarks of aging. Mm-hmm. Maybe explain to people what the hallmarks of aging are, which I don't mean like list them all, but the concept of them. Don't worry. I wouldn't put you on the spot for that. But the idea that has been proposed is that there are hallmarks of aging. And why is stating those not the same as answering the question that you, me, and everybody else is struggling with?
Well, this is kind of an existential crisis with me, because the hallmarks of aging came out in 2014 or 2013. And then I wrote another paper right after that called The Pillars of Aging, which is kind of the poor stepchild of the hallmarks of aging. And that was because there was an NIH conference and there were 7 topics discussed, and they asked me to write a review on calling them the Pillars of Aging. So I did. But even in that review, I had the 7 pillars of aging, but I connected them all with lines, because I don't really think that these hallmarks and pillars, which are like the pathways in the cell that are thought to be driving the aging process, inflammation, epigenetic changes, these kinds of things, they're all interesting to aging, and you can modify them, or they get modified if you slow the aging process. But what strikes me is how entrained everything is. So if you take an intervention like rapamycin that slows aging, it can impact all of the hallmarks. And so I think those are like outputs or ways you can look at aging, but they're not, nobody is really just targeted. The idea that you can target each hallmark and then you'll live forever is not going to work, because it's really the network that connects the hallmarks together. And to me, healthy aging is about maintaining homeostasis. It's about, maintaining a responsive network in your body that sort of keeps you in equilibrium, responds to the events that are happening during aging, the stochastic events, the damage that's happening, and it keeps you functional. And that network is highly malleable. You can influence that network by drugs or behavior. And if you do, you can derive benefit from it, and you can read it out as an improvement of all the hallmarks. It's not like one thing of exercise affects only this hallmark. So I think the hallmarks was good because it drove interest in the field. It's part of the reason a lot of investment came in the biotech sector. But it also is misleading, because I think the idea that aging is 12 different things and you just need to fix all 12 of them is completely wrong. It's really about your body knows how to function in a healthy way. It's about trying to maintain that and maybe improve upon it. So.
Do you look at the hallmarks, which I believe have been modified since the original paper and a few others have been added? Do you see a rank order or a seniority of them in terms of causality? So, for example, one of the hallmarks is mitochondrial dysfunction.
Yeah.
Now, one could say that mitochondrial dysfunction occurs independent of another hallmark of aging, which you've listed, epigenetic change. Alternatively, you could say, actually, it's the epigenetic change that occurs stochastically, and that is driving mitochondrial dysfunction. And if you reverse the epigenetic change to the previous epigenetic layout, you will correct the mitochondrial dysfunction. How do you think about the interconnectedness through the lens of causality?
Yeah, I think the primacy issue is a major one. I like the idea that mitochondria might have been one of the primary drivers. Also, every time we do an experiment, we keep coming back to inflammation. All of the interventions that extend lifespan reduce chronic inflammation, or almost all of them. And then every time we create a new biologic aging clock, which we're doing a lot of now in my lab, and we do principal components and figure out what the main driver is, it's always related to inflammation. And so I think there's something, it may not be, inflammation may be a response, but it's so central that a lot of the interventions, I think, are working by dampening inflammation.
But to your point, inflammation could easily be a readout state.
Yeah. But it's certainly, when you modify it, you get an outcome. So it's not just an endpoint that you look at.
How are you measuring inflammation? And maybe walk me through how you're doing it in different model systems. So are you studying inflammation in yeast?
Not so much in yeast, but you can study innate inflammation in worms and flies, because those pathways, the rudimentary elements of those pathways are there. And then in mice, you have both innate and adaptive immunity that you can study. And so we look at inflammatory cytokine panels and a range of other things in various tissues to see, how that's changing over time and how interventions impact that. And we do that in humans too in our clinical study.
And so what do you believe is the hallmark of maladaptive inflammation? Do you think that the hallmark of that is based on immune function, i.e. deteriorated immune function and or overaggressive immune function? Or do you think, no, the hallmark of that could simply be found in a cytokine profile that is not typical? I mean, this is maybe more of a technical question, but it's going to become interesting as we move our way into humans.
Yeah. Well, I think it's central to mTOR. One of the things we were one of the earliest people to publish was that what's happening during aging is that baseline levels of mTOR are creeping up. You can't turn the pathway off. And I think most of the interventions, sirtuins, mTOR, inflammation, it's not about doing anything super physiologic with the interventions. It's about restoring the dynamic range that you had when you're youthful. And mTOR is a great example of that, and I'll bring it back to inflammation. You need mTOR on when you wound your skin or you get an infection or you have a big meal in your liver, but you need it off the rest of the time. And when you're young, you're very good at maintaining that dynamic range. But what's happening with aging, at least in stem cells, is that the baseline levels of mTOR are creeping up.
People on this podcast are pretty familiar with mTOR. Yeah. We've had David Sabatini on many times, Matt Caberlin as well. But I just want to make sure that for anybody who's here, who's either new or forgot, let's take a step back. mTOR is so important to this discussion. Let's go back as far as we need to and explain mTOR.
Okay.
And talk about complex 1, talk about complex 2, talk about how one impacts the other. We're obviously going to talk about rapamycin in that context, but take as much time as you need to make sure listeners really understand why mTOR is so central across all of life that we've ever known.
Well, sort of our entry into the mTOR pathway was in yeast. This is Matt Caberlin and I. And so we were screening the yeast deletion set. It's a set of strains which each gene is deleted, and looking for one.
How many genes, by the way, in yeast?
They're about 5,000. And then some of those are essential, so you can't screen those. If you knock them out, the yeast are dead. But about 70% of them, the yeast are still viable. And so we were trying to find ones where when you knock out a gene, the yeast live longer. Surprisingly, there were like 300 genes that met that description. And that one thing I learned from that is that extending lifespan, at least in a simple organism like yeast, and there's also data in worms, is much easier than anybody would have ever expected. And we can talk about that.
But that's still a pretty cool tour de force in the 90s, right? You guys were doing this in the early 90s.
We did the sirtuin stuff in the 90s. We did the full genome screen in the 2000s when I was in Seattle with Matt. And so, but it was a lot of work, and we did it brute force. We had people sitting at microscopes dissecting yeast all the time. And I think there were 80 some authors on that paper. For a yeast paper, that might have been a record. But anyway, one of the main things we hit was the mTOR pathway, and downstream of it is protein translation. We hit a lot of things in protein translation. And mTOR is a nutrient-responsive kinase, so it responds to the levels of carbohydrates and also amino acids that the cell encounters. And so that fit into the calorie restriction data, which I'm sure you've talked about, that reducing calories can extend lifespan. So it seemed like it was going to be very central from the beginning in modifying aging. And I think that's proven true. Turning down mTOR signaling across a wide range of species extends lifespan. And I think the data in humans is also pretty, it's not fully validated, but I think that if you alter mTOR signaling in the right way, you can probably slow aging in humans too.
Now, why is it then that the first human brush with mTOR modulation shows up in the form of immune suppression?
Yeah, so I think that's a bit unfortunate in a way, So this rapamycin is discovered on Easter Island, and you've probably gone through the whole story of this. It almost died.
I feel like I'm overdue for a retell of it, but yes, it's one of my favorite. It's probably top 5 stories in science, right?
It's pretty amazing, actually, and I think I read you went.
Yeah, I wrote a
Easter,
chapter on it.
And you went to Easter Island too. Yep. I haven't even been there. I'm impressed.
We're gonna go back in 2016. So yep.
But this drug had the ability to kill bacterial cells. That was bactericidal. They'd start with, and then they discovered it had impacts on human cells, and but almost got thrown away, and then gets restored, and it makes a new life as an immune suppressant. And certainly, if you use it at high enough doses and you really dampen the ability to activate TOR, you can impair the immune system. And especially if you combine it with cyclosporin or some other anti-inflammatory or immune suppressant, then it's used for after organ transplant in those contexts.
And do you think that that it's that necessary combination with 1 to 2 other immune suppressants that allows it to shine? And I'm actually not aware of literature that looked at rapamycin or rapamycin in isolation as a potential treatment for organ transplant patients.
I'm not either. At high enough doses, it may have that impact, but I think the side effect profile would be too extreme. By combining it with other drugs at lower doses, I think you get a bigger effect. But I think the main thing for aging is that it's not immune suppressant, I think, at the levels that people are taking it for longevity, which is once a week, let the trough levels come down. And I don't think we're seeing immune suppression in that context, at least not above background. So,
yeah. And what do you think of the window we got into this idea of immune modulation and maybe immune enhancement with that type of a dosing regimen vis-a-vis the paper that Joan Manick and Lloyd Klickstein published about 12 years ago?
Yeah, I like those papers. I think there's definitely a nugget of truth in there, and I think it can protect from respiratory infections if used correctly. So I still think rapamycin is the gold standard for a small molecule impacting aging. At the end of the day, it may not be the best, but right now I think the evidence is still the best. And I think coming back to the earlier thought, when you have mTOR creeping up when it shouldn't be, that's driving chronic inflammation, and then chronic inflammation is continuing to drive mTOR. So it's this feed-forward circle of disruption that connects this nutrient pathway to inflammatory signaling. So I think that's one of the earliest events that's happening.
So let's go back to something you said a second ago, which is absent the equations, biologists have to rely on their intuition.
Yeah.
And so if we believe in the primacy of that deterioration, that homeostatic deterioration that you just described, what is driving it? Is this entropy? What is it that is changing?
Well, entropy has been used off and on ever since I started in the aging field.
It's just a lazy term we use because we don't have something better to say.
No, but I think there's a nugget of truth in that. And I think ultimately, and I think that, I work with Peter Fedichev at Gero, and I think he's probably the deepest thinker in the aging field in terms of understanding this process of aging at a mathematical level. My only role in working with him is I was a math major. Fortunately, I didn't try to pursue that. But I survived enough to get a math degree in college. So I'm kind of his whisperer or translator. I take what he says and try to help him reframe it in a way that the rest of the world can understand. He really thinks about things deeply. And I think his view on it is that really what we're talking about is resilience. You can imagine, from an image, you could imagine that, when you're young, you're living in this deep valley, and you do all kinds of crazy things. You get too much fast food, you get sick, and you diverge off the bottom of the valley, the lowest activation energy, and you're biologically, you look older, but you're in a deep valley. You keep getting pulled back to health. And so almost no matter what you do, at least in the short term, you're coming back into health. But if there are hills you can go over into what you would call failure states, which could be chronic diseases or could be some major functional decline. And that doesn't happen when you're young because it's a very steep hill, but these hills are coming down as you get older. And so when you diverge off the healthy state, it's harder to get you back. And occasionally you go over the side and then you're, in a kind of a frailty mode once you have a later stage disease. So the question is how to mathematically model that. And it's more of a dynamic systems type of modeling. And when you look at the large data, and I'm getting to the point where you're going to understand why I need the physicist. But when you look at the large data, it almost looks like there's a linear accumulation of damage. And so what we're doing now is we're trying to measure biologic age from large data sets like UK Biobank. And then we're breaking it down into principal components. And when you do that, a lot of the principal components don't track with aging. They track with sex or smoking or something else. The ones that track with aging, usually there are a couple of them. One of them is the primary driver, and it's kind of going up linearly. And that looks like damage. And that's probably the main driver of aging. It doesn't have to be damage. It could be stochastic events. It could be subtle changes.
But when you say damage, do you mean DNA? Like what damage are you tracking? DNA damage?
That's why I was trying to back off a bit because we don't. It may be a cluster of different types of damage. It may be stochasticity and not really defined as damage, subtle changes here and there that each on their own have tiny little impacts. But when they start to add up, they put stress on this network, and eventually it starts to break down. And so that looks linear. And the problem is that mortality looks exponential.
Yep.
And so if you model it into a mathematical equation that talks about these valleys going down, that's a linear change. But the chance the ball's going to go over the hill is an exponential change. And so what I like about it is you can fit human data into an equation that is compatible with Gompert's equations and exponential increase in mortality. So I think it's on the right track. I mean, there's probably a lot of changes. There's also another component usually in these biologic aging clocks that's age-related, and it's kind of oscillating. And it sort of oscillates around that first component going up. And I think that's why you see these methylation clocks are going up and down and changing and everything. And I think of that component as how well you're functioning at the damaged state you're at. So you have all these events that are going bad, and that's defining sort of your age. Maybe it says you're 50, but then you can be somewhere between 40 and 60, depending on, how well your behavioral patterns, what supplements or drugs you might be taking. And those things are going up and down. So that's why when you get sick, you look older, and then you get better. You come back down. That first driver is not changing. It's the second one that's oscillating. And most of the interventions seem to affect the second one, which suggests that what we're doing right now is, all right, I'm going to say this and then I'm going to qualify it. It suggests that what we're doing right now is sort of working around the edges. We're doing things that may have 5 or 10 years impact on health span, which, by the way, is a revolution if that's successful. I think that's a major breakthrough in medicine if we can give everybody 5 or 10 years of extra health span. But that these things may not impact maximum lifespan in humans, and they may not get us to 150 or 200. And the kinds of ways to get there may be totally different kinds of interventions. So we're thinking about that a lot with Peter right now. And I translated my research in. That translate is a bad term. I switched my research into translation about 10 years ago because I was like, I don't want to be retired and 80 on a porch somewhere and not have any impact on humans. But now I'm starting to think back a little bit to basic science, because I'm starting to think that the interventions that we need to develop if we really want to have the big changes are not being done yet. And we have to go back to some discovery science to do that.
So Brian, there's a lot you said there that's really interesting, and I'd like to unpack it both for myself and for the listener. The first thing you said, I actually am not sure I understand, is we have a linear process of, we're going to use the word damage loosely, and that over time, that is increasing monotonically and linearly.
And not alterably.
No, exactly. Superimposed on that, so we have, damage occurring this way. Superimposed on that, we have sort of cyclic, episodic, volatile change that probably explains a lot of the difference between two 50-year-olds that you might see. You might see a 50-year-old, they look great. You see a 50-year-old, they look like they're 75, right? Why? Or you might see it even within an individual. Boy, at 50, I looked horrible, but I got my act together. And by 55, I actually look like I did, 10 years sooner. Okay, so that's the superimposed curve. And you're saying, look, everything we're doing from a translational perspective, all the stuff Peter talks about, by the way, like wrote a whole book on this topic, is how do you impact the oscillation? Right? Well, if you sleep this way, if you exercise this way, if you eat this way, if you take this supplement, this drug, manage all of these factors, you are absolutely going to put yourself on the better wave here.
Right.
But you are not impacting this guy.
That's what I'm afraid is happening.
But then you said something a moment ago that I just want to make sure I understand. Is your explanation for why aging follows a Gompertz curve as opposed to a linear curve, is that all due to the superimposed wave that goes on top of the linear curve, or was there another reason that aging follows exponential Gompertz law?
No, I think I had conflated two ideas at once there. So let me dissociate that. You've described the wave better than I do, so we'll leave it at that. But I think that you're right. We're trying to get. It's almost like we're trying to get to the best state we can be at for the damaged state we're in.
That's right. By the way, I have never thought of it that way, Brian. I love that description, and it's actually what I say to my patients, because I get people that come to me and they say, Peter, I really want to live to 150. I'm told you're the guy. And I say, actually, I'm not the guy. I don't believe it's possible. What I believe is possible is 7 to 10 more years of infinitely higher quality life. And if that's not what you want, if you want something that is far in excess of that, you're going to have to go to somebody who's got proof that they can do different.
And I don't think there is any proof right now.
I obviously don't think there is either. But
I do leave the idea open that it could be possible. I think it may be feasible to do that, but I don't think anything.
It's just I don't see any evidence that there's anything that's doing it now.
Translationally, I agree with you. Now, I think the linear to exponential is the idea of the hills, right? So you've got a ball. If you do it at 2 dimension, you've got a curve that looks like this, and you've got a ball here, right? And the damage is causing the thing to come down. And so the chances the ball going over is actually exponential.
Yep. So even if you have a linear reduction in the height of the hills, the activation energy will increase probability exponentially over the curve. And by the way, I'm just trying to think through why that's the case. Is that the case because of, is it a V-squared problem in getting over the hill if we were to model it out as actual balls?
I have a graph. I have a model, a movie that really helps people that are not mathematicians. I think both of us have some understanding of the math, but a lot of people don't. And I think that, it's something like that. I think also it explains very well why treating disease doesn't work because you have 50 failure states you can go into.
Yep.
And each person, based on their genetics and their lifestyle, the hill to go over that failure state may be a little bit different. Sometimes person's not going to go over this one, but there's a chance you're going to go over a lot of different failures, right? And if you block one of them, say you treat diabetes or something, you're still going to go over the other ones, right?
Yeah.
And so the only way to really slow aging is to keep the hill higher.
Yeah.
So
no, that's both a beautiful model, a mental model for how to think about it, and yet still equally infuriating because I don't know why the walls are coming down.
Yeah.
Right?
Yeah.
Why are the hills coming down?
Well,
what is the fundamental reason? What's the particle reason for the wall height coming down?
I think the hills are resilience, right?
But even
resilience
still,
is the most important term in aging, and nobody understands it.
Yeah, exactly.
I think that, all right, I'll give you a sort of half-baked answer because it's all I can give you. I think that what's happening is this damage is impacting this network that's keeping you healthy, this homeostatic network. And it's in little ways here and there and here and there. And the network compensates for that and does okay. But when enough damage happens, you just can't compensate anymore for events that are happening. So when you get sick, you get some viral infection, or you fall down when you're 80 and break your hip, you just don't have that homeostasis pathways in place to allow you to recover and compensate for that. I mean, that's sort of the best answer I can give you right now.
So if you had to guess, and I'm hopeful that the listeners are with us, because this idea of the linear and monotonic increasing in damage is the thing that has to be addressed if we're going to make a step function change in human longevity,
so.
right?
Yeah.
So I like how you described it. We're really tinkering around the edges, right? Everything we do is tinkering around the edges. But if we fundamentally want to get to a point where maximal human lifespan is changed and health span is fundamentally altered, we have to bend the slope of that line.
Yeah.
So my first question for you is, what is the probability in your mind that rapamycin is doing that, based on what you've seen in animal models?
Well, okay. So first of all, if you look at something like a worm, I think that the modeling is very different there. Worms are just kind of already in a failure state. They're like designed to last for 2 weeks, and they don't have that homeostasis that humans have. And so you can get huge impacts.
Fooled by interventions there.
Yeah.
Yep.
In worms or other organisms that the pathway may translate to humans, but the effect size in humans is going to be much smaller. I suspect that's where rapamycin is, that it's going to give you a healthier period if you take it the right way. Or I don't think any intervention is going to affect everybody, okay, but a majority of people may benefit from that. But I think it's in the modest effect size, not in the change the hill.
Not in change slope.
Yeah.
Okay. I want to come back and talk more about RAPA and mTOR because you're, again, one of the few people, along with Matt, David, people who can really talk in depth about it. But let's now stay in the world of speculation. If you had to even imagine something that can change the slope of the line, i.e. can take, I guess we define maximal lifespan as the 90th percentile of lifespan. So let's just make a number up and say maximal human lifespan today is, I don't know,
105, maybe 90.
Something like that.
Yeah.
Okay.
120 is the
99.9999. Yeah,
Yeah.
exactly. So we're going to take 90th percentile human lifespan up by 25 years. If I told you, Brian, you might not be alive to see it, nor will I, but I have a crystal ball, and in the year 2100, 90% of humans will live to be 130. And now I say, give me your best guess as to what did this. Is your guess going to be small molecules, genetic engineering, epigenetic engineering? Like, just go down the pathway, or multimodal? It's going to have to be 10 different things. Like, this is just kind of like the fun sci-fi game.
Well, I think the scary thing is that linear accumulation, it does look like entropy, which in reversing the second law of thermodynamics is...
We don't have to reverse it. We just have to slow it.
Even slowing it is a challenge, right? So And if you think about it from a physics standpoint, which I'm getting further and further and deeper in a pool I shouldn't be in, I'm just telling you that right now. But if you
I'm giving you a life jacket. just keep treading water.
My motto, by the way, with the consulting I do is that I know what I don't know. It's not a good motto for consulting, and I've learned. But anyway, as you get into the physics of it, it's really about temperature. And I don't mean temperature in terms of the temperature in the room. I mean the energy in the system that's driving the changes or damage. And the question is, how do you lower that? And so maybe what you need to do when you're looking for longevity interventions is not looking for how long a worm lives, because the worm is dying for a different reason. It's already in the failure mode. It's about how to lower the noise in the system, and lowering the noise in the system might be a way of changing that slope. So that could be transcriptional noise. It could be anything you can measure as noise that happens over time. You might want to try to lower that noise. That's an interesting concept. Now that may also come with secondary effects that people don't want. I mean, so.
Right. There might be a retardation of growth and development early in life, and it might be one of those things where you don't want to touch this slope for the first 30 years of life. Or again, where's the point at which you want to intervene?
I think that temporal component is really important. It's taking us into a different concept, antagonistic pleiotropy. But people, and it is true that if you look at all the yeast mutants that extend lifespan, most of them would not make happy yeast in the wild. They slow growth, or they do something else, affects some property like mating that is not going to make for a yeast that survives through natural selection. But if you put them in a lab, they can divide more times. And so a lot of long-lived mutants have fitness cost. And so the question would be that if you target the noise in the system, which is a completely different way of thinking about interventions, what will the fitness cost be with that? And you're right, maybe you can get around it by temporal things. Like the mTOR pathway, you probably don't want to impact as a child. But as an adult, it's more important early in life than it is later in life. It's only important at certain times. So if you impact it the right way, you can get the benefits without the cost. And maybe that's possible in these interventions. But we're so early, I can't even with any confidence tell you what kinds of interventions would have that impact. I will say one other thing is that I think reprogramming is potentially a way to mitigate some of this entropic change, because if you can replace the cells, you with new cells, those new cells may have some of the damage because they come from the old cells, but they will probably get rid of a lot of the damage too, and so that may be a way of changing the slope. So like reprogramming, which I think is still very early stage, that may be.
So as a thought experiment, if you took if I could clone you right now, or you had a twin, let's just say you've got a twin.
One of me is enough in the world.
But you're in Singapore, so we're going to have a North American version, we're going to have the Singaporean version. So we have two of you, and in one of you, we're just going to act as the control. We're going to give you some vehicle. In the second one, let's just assume I can use the fidelity of CRISPR to revert your entire epigenome in every cell of your body to what it looked like when you were 20. And anytime it gets out of whack, I smack it right back to 20-year-old Brian. Epigenome only. Not genome, not proteome, not nothing. I don't change anything but epigenome.
Well, those things are going to change.
They're going to affect everything else. But what is your guess as to the difference in lifespan and health span of those two versions of you?
That's an interesting question. I think there would be a difference for sure. I'm not sure it would be a huge difference.
You don't think it would be huge? That's interesting.
I'm not as sold on the primacy of the epigenetics. Certainly you can modify.
So what would go wrong, right? So if you look at, and by the way, I don't know the answer, of course. I'm just kind of thinking through data that I've seen, right? So if you look at epigenetic code for two different hepatocytes, liver cells, one from a 20-year-old, one from a 50-year-old, and I tell you which one's 20, which one's 50, and then I show you a bunch of others. You can always tell which one's the older one, which one's the younger one.
I can probably tell that from mitochondria or a lot of other things too, though.
Yes. So the question is, do you just say, is your belief system that just because you revert the epigenome back to what it looked like when it's 20, it's not going to change gene expression enough to move the needle?
I think it'll definitely influence gene expression, but there's also DNA damage that's happened. There are mitochondrial changes. The question we're asking is, if you revert that, how many of these other things can we fix and restore?
Yes.
And that's the unknown answer. I suspect you would have a significant impact on those things, but not fully restore them. I think the question of the primacy of the epigenome is an open question. Nobody knows the answer to this.
How testable is the hypothesis? How would you design the experiment to test that?
Yeah, I think that's difficult because if you want to do it in a very direct way, you really need to modify the factors that are controlling the epigenetic regulation. But there are a lot of factors doing that. It's not just DNA methylation. It's histone modification. There's nuclear packaging and nuclear lamins, which are linked to aging as well. And there's not just one pathway to change. So I think from a real life standpoint, it's hard to think about how you would do that effectively. Certainly, I think people have really jumped on it. And maybe when I'm talking about aging clocks at some point, people have really jumped on this idea of epigenome being the driver of aging, because you can get a biologic age by measuring the DNA methylation changes across the genome.
Well, I think they're asserting that that's the case. I don't see any evidence that that's the case.
We can come to that.
Yeah. We definitely want to dig into this.
You can get to that same point by measuring the proteomic changes, by measuring the microbiome changes, by looking at facial structural changes. Jackie Han's got great data on that in China. So anything in a human where you have a deep enough data set that's enriched enough, and you have samples across a wide enough age range, you can make a clock that predicts their age. And the facial clock is about as accurate as the methylation clock. So I think that a lot of people have jumped on this methylation or epigenetic bandwagon, but they're taking association and causality, and they're making a big leap there. Now, we know that you can modify epigenetic factors and extend the lifespan of yeast and worms and flies, maybe even mice. So it does have a role, but you can do that with senolytic factors or nutritional regulators or, calorie restriction or a lot of other things. It's not clear to me that that's a bigger effect than you're going to get from targeting these other interventions. But to be fair, none of them are completely reversing things either. So they're not addressing your question directly.
Yeah. Do you believe that and this is just, again, we're in the philosophical, I'll bring it back to reality at some point today. Do you think that immortality is impossible? Unless we define it through kind of like AI copying your brain. Let's put physical immortality. Do you believe that is impossible? Well,
I like to tell people that I'm immortal because I think the mindset it gives me is a very healthy mindset for me. We can come to, you talk a lot about the emotional aspects of aging in your book. I love that chapter. We can come to that later. But I don't really believe it's true. I think the odds that you could achieve that level of change in aging is non-zero, but close. So I'm skeptical that can be done. I think it's fair to say, but I wouldn't rule out the possibility. Of course, nobody's ultimately going to be immortal because you're going to get hit by a bus sooner or later. Yeah. But what you're really talking about is being immortal in terms of dying from aging.
Yeah, I guess what I'm really saying is, can one ever get to the point where resilience is high enough that you cannot die from disease?
I have seen nothing so far that suggests that's possible. Okay, but that doesn't mean it isn't possible.
Yeah. And then that gets even to physical frailty and sarcopenia and things like that, where even when we see centenarians and supercentenarians, their frailty is still pretty remarkable.
Meaning?
Meaning, like, they still look pretty feeble and frail. The age adjusted, they're great. Yeah. But at the end of the day, Like when they're 110, they still look like someone who's in the final years of their life, just as someone would if they were 84, 85.
Yeah. I mean, I had two grandmothers that lived to almost 100. One died at 99 and the other 101. And the one at 101, I would say that at 90, she was driving at 95. I think she quit driving. She bowled a 238 game at 93. She looked like a
at 93.
Yeah.
But so that's
she
my
looked
point.
like a 70-something-year-old.
Yeah,
at that
exactly.
point.
My point is she just had a phase shift of 20 years, but it didn't undo the inevitability of that decline.
Yeah. No, I agree with that. I think that, but getting us to 100 is a good goal, I think.
I agree completely. Do you think that, again, we're spending too much time worrying about finding immortality, escape velocity, understanding the core of aging, when maybe we should be spending more time on how do we preserve health span in the last decade of life? Why is it that most people in the final decade of their life are physically too frail to enjoy life, are cognitively just, even absent Alzheimer's disease, they're just not cognitively sharp enough. They're in pain. They're fracturing their hips. They're not doing what gave them joy through most of their
life. I agree
life.
with that. Yeah. I think that we should fund aging somewhere closer to the level we're funding cancer and answer both of those questions at the same time. I think one of them is a translational question about how do we slow aging as much as we can right now and improve the health of the population as much as possible. And the other one is a basic science question. Can we stop aging? Can we reverse aging? No one can. If anybody tells you they have the answers to that, they're lying to you or they're lying to themselves. Okay, we don't know, and I think that. But it's maybe the most important question in biology, and we should be throwing money at it. So we've seen all this money go into like the private sector side, biotech companies, supplement companies, longevity clinics, and on and on. And I think that's great, by the way. And I've spent a lot of my time working with those groups because I think it's important. But we're not seeing the academic funding that's going into the basic science of aging and longevity. And the big questions still that you just raised are still not answered. And so I'm changing your question to a plea for more funding. And unfortunately, the kind of funding that supports that is usually government funding, foundation funding, and that's under major threat right now. So it's I'm really worried that we're not going to answer those questions.
Well, it's funny. This was a discussion that came up on a longevity roundtable. I think most people, myself included, were really surprised to hear how disparate the funding differences are, and how if you could if you could reallocate 10% of funding from the disease-specific pools to the age pools, it could have an enormous difference based on how little the funding is. Yeah. So when you think about the big chronic diseases, cardiovascular disease, cancer, dementing diseases, and metabolic diseases, right? Those would be the big four. I've often maintained that the least inevitable of them is ironically the one that is the most deadly today, which is cardiovascular disease.
Yeah, I agree.
The atherosclerotic diseases, so cerebro and cardiovascular, ironically the most preventable.
Yeah, I think so too.
Because we have the best understanding of what causes them, and we couple that with the most tools to prevent them, whether it be tools to combat hypertension, dyslipidemia, et cetera.
And they're responsive to lifestyle modification.
Yeah.
I mean,
which of those major diseases, of the other three, dementing, cancer, metabolic, do you believe is the most inevitable to our species?
I wouldn't put metabolic in that category for sure, because I see that more like cardiovascular.
I agree. Yeah. So of the other two, cancer and dementing or neurodegenerative diseases, which one is just seemingly inevitable?
We don't know enough about dementia to answer, but I will say that cancer is a little bit different than these other diseases, I think, and it's not and it may be less modifiable by longevity interventions. Dementia, we just don't know. My guess is it's highly modifiable too, but there's not enough data to be sure of that, like there is for metabolic and cardiovascular disease. But cancer is an accumulation of mutations, so it's a more defined event that's happening. It's also a impact on the immune system that's different a little bit than normal aging. So it may be less approachable from a longevity viewpoint.
Yeah, it's funny. That's exactly my view, that cancer is the most inevitable of these diseases. Yeah. Do you think that the inevitability or the age-related component stems more from the accumulation of mutations or the weakening of the immune system?
It's probably both. I mean, you don't get to cancer without the right mutations happening. But I think we're learning more and more that the immune system is playing a major role in it, and that we can see that very clearly from the interventions that improve immune function, and they're having a big role in certain types of tumors. But I think that's going to be true for Alzheimer's and dementia as well. We've completely underestimated the role of inflammation in the immune system and those diseases as well. And they may be the primary drivers. You know, I kind of I'm very frustrated by some of these fields. One of them is Alzheimer's. You know, I kind of feel like, you know, one of these Alzheimer's researchers, they're going to die at some point of 90, and on their tombstone, it's going to be like, you know, major accomplishment was to completely remove plaques from the brain. Died of Alzheimer's at 94. You know, there's been so much focus on one or two mechanisms of disease that we spent 30 years not studying the others, which may be more important.
Why do you think that? I mean, I write about it in the book, but I'm really curious as to why you think that's happened, and because unfortunately, that's not an isolated incident in science. So why do you think it's happening in a field where the results are otherwise so dismal?
What's the saying? Scientific progress happens one funeral
at a time.
Yeah, I think that's part of it. You know, you get people that have successful research programs, and their postdocs get hired in all the jobs, and so when you take a field and it grows from a small field to a bigger field, everybody can draw their lineage back to 4 or 5 different PIs, and so whatever models, and those PIs get really focused on those models, and they see that as their ticket to prizes and things like that, and so then you focus on a subset of the disease mechanisms at the exclusion of all others. And I don't want to single Alzheimer's out. I think a lot of diseases meet that category, but it's unfortunate because what we're realizing is that, you know, there's a lot of factors that contribute to any disease. And I think longevity may be an interesting way of looking at it. Like, I think it's better, like, I'll take a mouse, okay? We've tried to make Alzheimer's models in mice, and they don't prove that informative. Why? You're creating a disease a mouse doesn't get genetically in a young mouse and comparing that to a natural disease in an old human. I think you learn more about Alzheimer's if you look at the brain neurodegenerative changes that happen in the mouse normally with aging. The downstream things are different, but the drivers may be very similar to the ones that are driving Alzheimer's. And that may be a better model of Alzheimer's than trying to artificially create something that a mouse doesn't get. So that's what I think aging is helping change that perspective, that, you know, the drivers of aging, I think, are very similar between a mouse and a human. The downstream events can be different, but the drivers are what we care about, right? So
let's go back to rapamycin for a moment. Do you believe that the primary effect of rapamycin is tamping down maladaptive inflammation through, obviously, the intermittent blunting of mTOR?
I think that's one of the major things, certainly. You know, that there's good evidence for enhancement of autophagy. There's good evidence for changes in protein translation, and those things are not mutually exclusive to inflammatory changes anyway. But I think those are the three things that we have pretty good evidence for. But I really think that all of these interventions that we're looking at are restoring dynamic range. They're not. It may take supraphysiologic changes to change that linear line, right? But I don't think that's what we're looking at right now. We're restoring things that happened when you were young.
So given that we're not likely to have human clinical trials of rapamycin that study aging, for the simple fact that we don't even know what an aging biomarker is, we're going to largely be extrapolating from animal data if we have to make decisions about humans using rapamycin for geroprotection.
All right, I'll push back a little bit, and then I know where we're going with this, but we're doing a study like this in Singapore on humans. It's a 6-month intervention with rapamycin. And we're looking at as many different parameters of age. We're not doing disease. In fact, we're not taking people with disease.
Yep.
We're taking people that are 40 to 60. They may have a precondition for a disease, but they don't have anything
healthy.
that would be defined as a disease.
Okay.
So it could be, high glucose, but not diabetes, and so and then we're looking at changes in a wide range of different biomarkers, clocks, which.
So tell me a little bit more about the study. So how many subjects?
We're doing these, I don't remember the exact numbers. It's somewhere around 150 to 200. So it's not huge.
And how you're dosing it?
Intermittently.
Once a week?
Yeah.
How much?
Yeah. I think it's 5 milligrams is what's in the protocol. It's being run by Andrea Meyer, which is one of my collaborators in Singapore.
Got it. So 5 milligrams of rapamycin once a week for 6 months. And then let's go through all the different measurements.
Yeah, so a range of clocks, inflammatory cytokine panels, functional measures, pulse wave velocity, DEXA, strength measurements, cognitive measurements, and I'm still missing a couple of them. But
do you expect to see changes in strength or cognition or things like that? I mean, do you worry that those are kind of the wrong outcomes to look for in a 6-month study of people that are young?
Yeah, well, I think that, this comes back to what do you measure? And this is where I knew we were going with this. I don't think we know. I mean, certainly you can change those parameters. I think if you exercise, you're going to change your strength parameters for sure,
If this was a 6-month exercise trial, fill your boots. Yeah.
I think 6-month exercise trial might also change the cognitive parameters. It's possible.
Yep.
So I think it's feasible.
Look, a 6-month sleep correction trial would undoubtedly change cognitive parameters.
Yeah. So I don't think it's unreasonable that a drug could do these things as well. I think rapamycin is complicated when it comes to muscle, and I know that partly because I kind of feel like I'm going to be non-scientific for a minute. I've become my own best model organism. So I try all kinds of different things on myself now. I know it's N=1. I'm not sure rapamycin and skeletal muscle, without exercise, I'm not sure what it's going to do. One of the things I notice is when I take rapamycin, if I do like a hard run, I'm a runner. I've gone to more lifting the last 3 or 4 years, but I've always been a runner. I don't have good runs within 24 hours of taking rapamycin, and it may be because you have to activate mTOR in a context or something like that.
Sorry, if you take Rapa 24 hours prior to a run.
Within 24 hours of running. Yeah.
Got it. Now, what about if you take Rapa after a run? Is your recovery better? Do you feel less?
I don't have a sense of that. What I do know is if 3 or 4 days after I take Rapa, I have really good training. So I think what's happening is that maybe in that short window after you take it, you can't activate the pathway enough. But in the long term, what you're doing is dampening the basal signaling, and you're getting the better dynamic range. So if the trough levels are low, I think, but that's me. But that's just something
I'm going to experiment with that, Brian, because I always take Rapa the same day of the week.
Yeah,
and my workouts are always the same. Like I do the same workouts on the same days of the week. I'm gonna do an adjustment on that and see. And what it does.
I didn't try it with resistance either, so I don't know what it's going to do there. So.
What tools do we have to measure autophagy in humans?
Well, you can pull out blood cells. We're talking about the limit. I mean, we're talking about the limitations of what you can get from a human, right?
Yep.
Blood, saliva. That's where we're going, I think. And so you can pull out blood cells, and you can look at, white cells and see, whether autophagy pathways are induced or not. You can take muscle biopsies. We don't really like doing that in our clinical studies because it makes it harder to get volunteers. I think if you do muscle biopsies the right way, they're probably not that painful to people, but people have that perception, and we need healthy volunteers for our studies. I would love to look at muscle. I think that, autophagy is another one of these dual-edged swords, though, right? You don't want autophagy on all the time. You want it on at the appropriate levels at the right time. And if it's on all the time, you're going to get muscle atrophy probably. So it's, again, it's about dynamic range.
What would you need to see in this study to feel that rapamycin is geroprotective? Because my concern, I suppose, would be You're not going to see a difference in DEXA. You're not going to see a difference in physical performance. You're not going to see a difference in cognitive function. You might see a reduction in certain cytokines, but not all cytokines. I forget what the other markers that you said were, but I guess my concern is we're not able, since we can't measure aging, we're not going to see enough of a signal. The other, you mentioned epigenetic clocks. In an ideal world, that would be the perfect tool to measure them, except for the Caberlin experiment, which I'm sure you're familiar with.
Well, but yeah, I agree with, I bring Matt to all my conferences now because I use that slide.
Where he did like 8 of them at once? Yeah, I mean.
There are multiple issues here. The one issue is, are the consumer testing companies, do they have a standardized enough protocol that it's reliable? And I think I'm skeptical that that's the case. I think they're—
And you're doing in-house measurement.
You're
not using anything consumer.
We can control everything. So we get around a lot of those problems.
Just for the listeners so they understand what we're talking about. Matt Caberlin bought 4 of the top commercial tests, did them in duplicate simultaneously. So it took 8 tests at the same moment in time. And he has a funny graph that shows how pathetic they are. Not only do none of the tests agree with each other, the identical tests rarely agree with each other. Yeah. So just if you're listening to this and you want to go out and get a commercial test that tells you how old you are biologically, reconsider it.
Okay, I want to tell you we have the potential for a better clock in a minute. So let's put a pin in that.
I want to talk about that, yeah.
So consumer-wise, I think there's concerns. I think
what are you using as a control when you talk about, again, given how inflammation and epigenetic change might be the only 2 signals that you find here, and again, this is just me being pessimistic nanny, but this is just my prediction. I do not think there will be a finding in any of those other measurements, but you might have a chance with inflammation and epigenome if you're measuring.
Maybe pulse wave too.
I just think, here's my problem with pulse wave, Brian. It is so user dependent in terms of the technician who is doing, like, we don't use it clinically at all because we think it's a useless test. Because like I think the carotid intimal thickening is a useless test, and that's an easier test to do because unless you have a tech who basically has a PhD in how to do vascular imaging, and they're the only one that does it every minute of every day. If my patients come in with a CIMT, I open a bird cage, I take out the poopy bottom paper, I put their CIMT in there, I close the bird cage. That's how useless it is. So I just worry that all of those tests are not going to, they're just going to be noise, no signal, but these other 2 might have signal. So then what's your control for accelerated aging or something else? In other words, it'd be really interesting if you did a 6-month parallel fasting trial where if you took people and you rendered them hypocaloric, you put them on some draconian 60% calorie diet for 6 months, where you really think you would tamp down inflammation and autophagy, and if anything's going to reprogram the epigenome in 6 months, you think that would be it. That would be a very interesting control, even if you had a fraction of the number of subjects.
Yeah, I mean, we're doing multiple. The first study we did was with a time-release version of AKG with PDL Health.
Alpha-ketoglutarate?
Yeah.
Okay, I'd like to hear more about that as well.
That's finished, and we're just analyzing data, so I can't tell you much data, but we finished the trial. We did 6-month intervention and 3-month follow-up. So we want to see if there are changes, do they maintain if you stop taking the compound? Okay. Because in mice, oddly, they do for aging, but I'm skeptical that's going to happen in humans. So we're doing multiple studies. So
this is sort of like your own ITP. They're not all at the same time, but we will be doing the same study over and over and over again with different interventions. Yeah. It's like I was just saying, your ITP equivalent in humans.
Yeah. And by the way, I don't know if 6 months is long enough. I don't know if we're doing the right tests. I don't think anybody knows these answers.
What is the cost of doing that experiment you just described?
It's about $1.5 million Sing. So that's $1 million to $1.5 million US.
Yeah, this is really interesting. And not that I'm here to do this, but it's just I get asked so much by people, where can I put money? Where can I put money? Yeah. So I'd like to make sure that people who are listening to this, who are thinking about, hey, how do I fund insanely high levered research, This, to me, strikes a great way
Thank
to be
you.
funding this, right?
We would love to have that because it's really hard to raise money for this still.
Yeah, but if you think about it, if you're sitting there and you're listening to this and you're saying, look, I'd like to put $1 million to work on something that would dramatically change, adding a decade to life, you're way better off putting, in my opinion, you really are better off putting money into this type of translational research than you are into basic science or into pharma research that tends to be a little bit more you're not going to move the needle.
Pharma's still not doing aging. I mean, they're thinking about it. They're starting to, but they've been saying that for a long time.
Yeah, no,
too.
this is great. As you can tell, I have strong opinions on ideas for how to do some of these experiments.
Yeah, no, that's great.
So let's talk a little bit about alpha-ketoglutarate and why. Walk us through the rationale for how that came to be something that you would put through this type of rigorous program.
Yeah, so company, full disclosure, that I'm involved with, PDL Health, they have a product, Rejuven now. They came to us many years ago at the Buck and working with Gordon Lithgow and I, and they were like, let's screen natural products that would have an impact on aging because, the mindset of the CEO of what became the CEO of this company was that I can't get drugs approved for aging at any time in the near future. Let's work with natural products and also
things that the FDA just calls grass out of the gate. Okay.
And let's look for combinations of things that work together because we can get IP around combinations. We can't get IP around single natural products. So we screened a lot of things in worms, and AKG came out as one of the best things in worms. And then we started testing interventions in mice, and that's led now to we're testing. Like we do 4 or 5 different intervention studies in mice every 6 months now, not just with natural products and not just for this company, but we built that into our own kind of ITP in mice too, although we do it differently. But anyway, AKG came out as one of the biggest effects. And then in the mouse studies, we found that for male mice, there was a combined effect with vitamin A. And vitamins are an interesting discussion too. And there was also a combined effect with both sexes with vitamin D. And so that led to this product Rejuven, which is AKG, time-release AKG. Very important because otherwise it's like it goes away in 5 minutes if you don't have a time-release version.
Tell folks what alpha-ketoglutarate is. It shows it's a part of the Is it part of the Krebs cycle?
It is. It's the TCA cycle, the Krebs cycle, central metabolite involved in hundreds of reactions. It's a lot like NAD.
Yep.
They're doing different things, but they're both central metabolites. They're both going down with aging in organisms.
Yep.
And the idea is supplementing them back up would be beneficial. And so when we did that with AKG in mice, we see about a 5 to 10% increase in lifespan, but a dramatic increase in frailty. So when we
meaning a
decrease in frailty.
Yeah, I know what you mean.
Yeah. So the mice, I would argue that you're extending
squaring
healthspan.
the longevity curve as we talk about it. Yep.
Yeah. So if that translated to humans, it would be a big impact, and so that's what led to the human product.
By the way, I think that if you did not extend lifespan by a day, but you just improved health span, that's a home run.
Yeah, I agree.
For most people, that is all they actually want.
Yeah, I agree. I think if they get that, they'll want the other two. But that's But
But never the reverse.
No,
nobody wants more lifespan.
The reverse is what we're doing now.
I know. That's called medicine 2.0.
Yep. But yeah, I completely agree with that. So, we're still excited about this product. There's been about AKG and especially the time release version, and there's been some studies that have been published, one of which we helped analyze the data for using rudimentary methylation clocks, showing that they reverse it reverses aging by a few years. Again, it's that oscillation thing we were talking about earlier, I think. And that's why we wanted to do a controlled placebo double-blinded clinical trial at the university. In that case, we're just testing the time-release AKG. We didn't include the vitamins because we're trying to get some mechanistic information. We don't want confounders in there. But I think as the data is still pretty good on AKG, that it's going to have an impact in people.
So that human trial has been completed. You're evaluating the data. You're going to have to see a signal. But are you in each of these trials? Do you do the same measurements, the same outcomes?
Generally, yes. But we sometimes modify the primary endpoint because we want to choose a primary endpoint that's most likely impacted by the intervention. In a way, it doesn't matter that much because we're measuring as many things as possible anyway. Of course, we learn over time as we do things, so we add things or take things out that are not working well, that sort of thing. So,
did you guys do a study on urolithin A as well?
We haven't published it yet, but I am happy to talk a little bit about it. We don't have human data, but the mouse data is really good on urolithin A. I'm a believer. It worked in our hands this is why we haven't published yet, because when we did it, it dramatically reduced frailty in male mice, but not females. And so we're repeating that because there may be sex-specific differences, but we're the first ones to see that. So we want to go back and see what's going on.
Now, I feel like I've written a newsletter on this where I came down on the side of this isn't doing anything. Am I mistaking this for a different molecule? Is this the one that in theory enhances mitochondrial function? Is that the thought,
the idea is that it enhances mitophagy, so turnover of damaged mitochondria. I'm not sure I completely agree with that. We do see that in cell culture, but we also see mitochondrial biogenesis. And so these two things are connected, right? If you induce mitochondrial biogenesis, you'll also induce mitophagy. And so it's kind of, I'm not sure where's the chicken and where's the egg in this, but it does seem to induce mitochondrial turnover. But we also find other pathways. When we look at a molecule, if we don't know enough about it, like rapamycin, we know it binds to mTOR. Urolithin, we don't know what it does. And AKG also, we know a lot of things it can do. We don't know which ones are relevant for aging. For urolithin, we went back and did this screening assay, the proteomics thermal shift assay, to look for binding partners for the compound. And we have some.
So tell me about the history that led to using it.
Well, for us, I mean, Johan Ower published this data that it was slowing aging in mice, and that's led to a lot of research. So we weren't the first ones to study urolithin. And a lot of what we do is testing interventions that come from other labs, because if I'm going to do a human study, I want to at least see it repeat in my hands in an animal first.
Absolutely. Yeah.
If it doesn't repeat in my hands, that doesn't mean it doesn't work. It may mean the conditions are different, but I think it does. If it does repeat, it makes an argument for robustness. And so that's why we started urolithin. We also see positive effects of spermidine and glycine and other things. So we didn't make the initial discovery on urolithin, but
so we don't know the mechanism of action.
Well, they would argue it's increased mitochondrial turnover, but we don't
have a target.
We have targets we haven't published yet.
Oh, you do?
Yeah,
okay.
Yeah.
Did you guys discover the target?
Yeah, we've got a couple new targets that we haven't published yet, so that could explain some of the effects.
And have you studied this in humans yet?
No, not yet. That's planned, but hasn't been done yet.
This might be actually one of the ones where I would say, unlike in the RAPA trial, you actually want to come up with primary outcomes that are quite different here. Yeah. This is where, for example, I'd want to see fat oxidation. I'd want to see, what we talk about is zone 2 efficiency. Yeah. I mean, if this is indeed improving mitochondrial function, I'm not aware of a better test of mitochondrial function.
Yeah, no, I agree with you. And that raises the point of whether we should couple these interventions to exercise. And
that one for sure
that
should be
adds
coupled
complication, but it may be worth doing in this context.
And not to get in your business, you must muscle biopsy these people.
Yeah, I know.
You're going to have to get athletes. You're going to have to get people that are willing to get a little punch biopsy of the quads. Yeah. Because it's too important to understand what's happening.
I think a lot of these supplements, I'm going to go back to an N=1 story here.
Yeah.
I think a lot of these supplements are impacting exercise. And so it's kind of a win-win. You take something, you exercise better, you drive benefits from that exercise. So it's kind of, you win twice with some of these. And I'll tell you what happened recently. I've been very skeptical of NAD. Not that going down and restoring it won't be good, but I'm skeptical it's going through sirtuins. I think it could be doing a lot of other things. But I'm also skeptical that NR and MN are really changing NAD levels that much. And our mice don't really respond in our studies with NR and MN. I've never noticed anything taking NR. Again, that's just one person, but that's me.
And when I last looked at this, and I know I'm going to get a lot of hate mail, because I always get hate mail when I talk about this, the only study I've seen in humans that shows a real benefit of NR was in patients with ALS that had the patients in the NR group had a longer time before requiring ventilation than the patients on the placebo. Yeah. Is there another study I'm missing?
Well, there have been healthspan studies arguing improved healthspan, but
by what metric?
I can tell you we've done them as well, and we do see very subtle changes within our that are aspects of the frailty measurements we do.
In mice or in humans?
In mice, but it's not enough to really convince us it's statistically significant. So I wouldn't be shocked if there's a tiny effect, is what I'm saying. But
okay. And why do you think that is? Do you think it's that NR and NMN are not efficient vehicles to generate enough NAD?
I think that's one thing for sure. But well, let me tell you the rest of the story, because I've pretty much given up on the pathway. And a company that came to me and wanted to test one of their products, and this company is called IX Biopharma anyway. They have a new product that's sublingual NAD. So they specialize in technology for sublingual delivery, and they make other things too. Viagra,
so we'll
sublingual.
just explain to folks why that matters. You can't take NAD orally because it just gets destroyed. So typically people take NAD intravenously. But if you take something under the tongue, you get this magical property where it dissolves and it enters the circulation without passing through the digestion and obviously the liver where these things get chewed up.
And you can do this every day, right? IV.
I mean, lots of drugs are given in this manner, right?
IV is just not practical on a repetitive basis.
How many milligrams of NAD are they?
100 milligrams. And the one I'm taking also has epigenome in it, which is a CD38 inhibitor. So CD38 is a consumer of NAD. So if you block that enzyme, this is another natural product. If you block that enzyme, then you also effectively increase NAD.
Okay.
And so I was taking that along with the Rejuvenate, which is the AKG plus vitamin A, and also the B complex, but mainly AKG effect. And when I take them together, I notice this acute effect on my exercise performance. So I'm running, my heart rate goes up. My respiratory rate doesn't go up as much. It goes up a little bit, but normally if my heart rate's at 150 or 155, I'm breathing hard. I'm breathing closer to normally when I take these two things together. I've gone off, I've gone back on, I've taken one off.
Is your rate of perceived exertion tracking more with your respiratory rate or your heart rate?
It's tracking more with my respiratory rate. I don't perceive that I'm exerting. I run faster when I do it.
You know what would be so cool to see, Brian, is measuring your lactate levels at a fixed heart rate under those two different respiratory rates, but under the same load. So do it on a treadmill just to make it unambiguous. I'd be super curious to see a lactate performance curve.
I should do that. I don't think you can imagine this. I've got, things telling me these two parameters, right? So, it's really...
I'd like to just quit my job and do nothing other than these experiments.
Yeah, and like I said, this may happen for no one else. I don't know. But for me, I can go off. It sort of goes away. It's starting to not go away now because I've been it's improving my exercise performance, and now I'm getting more fit. So it's getting harder to see the effect. But it still comes and goes when I go on and off either the AKG or the NAD.
The other thing I'd be interested to see is, let's pretend off drug, heart rate 150, RPE 7, velocity X. On drug, I want you to go back to the same RPE, let heart rate go higher and let velocity go higher. And I'm curious as to how long you can sustain that relative to what you were doing before. In other words, like, does some other system get in the way that ultimately reduces your capacity?
I can't answer, but the reason I noticed is that I'd taken it 3 days. I didn't even think about it. It's like every morning I do it. I was on the treadmill and I'm running, and I'm running. I keep pushing the speed up, and I'm not getting out of breath. And I was going to run 5K. I ran 12K, and I still didn't feel that tired at the end of it. And then I started thinking, how did this happen? Because because I know how my body performs normally.
So is this molecule in trials yet with humans?
It's not even have to be in trials. It's a natural product. So it's
no,
good.
but I mean,
it's on the market.
Yes,
but,
yes. But if for those of us who want to actually know if it works.
Oh, come on.
Yeah. Those of us who believe in this pesky thing called evidence.
Yeah. I'm getting past that now. I'm joking. There have been a lot of animal studies, and they can show that when I'm not even sure this is published. I think I can say this. When they add sublingually in a rat model, you have to anesthetize the rats to get the sublingual delivery.
Yeah.
That you see it incorporated very highly in red blood cells, the NAD. This was just with the NAD, not epigenin. And so the argument, there is literature to support this, and it's not my field, but I'll just say what it is that there are channels that can take up NAD directly in certain cell types. There is literature out there for that.
I had Josh Rabinowitz on the podcast, and he talked at length about this. And I mean, what I took away from that discussion was intravenous NAD will work.
Yeah.
The question is, what's it working for? And so would your belief be that the effect you experience here should be mirrored by what somebody experiences with intravenous NAD, notwithstanding the limitations of the frequency that they could do it?
Yes, I think so. But the dose is so much higher on IV NAD. And it could be too high. I don't know. But in theory, yes. And but this is so simple. You just every morning it's gone in 30 seconds. And so I think that and for me, again, it's enhanced by the AKG too. So you're adding two metabolites that are both going down with aging, that are both involved in hundreds of different, they're giving you cellular metabolic flexibility. I think that's what they're doing. I don't think it's one pathway they're activating.
And is the alpha-ketoglutarate with it's vitamin E or A that it's combined?
For a male, it's vitamin A, and the new product has some B complex in it as well. So.
And these are commercially available products?
Yeah.
And you're about to publish on them in humans?
The Rejuvenant has already been published on the AKG.
By itself?
Uncontrolled, not placebo-controlled methylation study of users. Okay, so take what you want out of that.
Not much. I'm just going to say.
Well,
you can talk me off my.
No. I don't want to try to. But I will say that I think Community Day is valuable, and I think if you go back and look at the paper, we didn't do any of the analysis. We just analyzed the data. But if you go back and look at the paper, I think we were very clear in that paper, even in the abstract, of the limitations of the finding. So I think that for community-based data, it's better to have it out there and published, but. You want authors that are willing to be honest about what the data says and what the data doesn't say. But the study that's been completed is just AKG, time release, so none of the vitamins, and it's placebo-controlled and as many parameters as we can measure. So hopefully it'll show something. Okay.
And then you're doing the one with vitamin A and E?
No, we're not doing that right now. We'd love to do that, but we financed this study ourselves. The only thing the company did was supply the time release AKG. And so it would be good to go back and look at the actual product and combined with this NAD now. But I just sort of figured that out myself 5 months ago. I don't even have any animal data that's true. It would be interesting to go back and add these things in animals, but you can anesthetize a rat and do sublingual delivery one time and measure the PK/PD.
Yeah, but you can't do performance.
All the time is going to be a nightmare.
You mentioned spermidine a moment ago. Let's talk about that. It's getting quite a bit of buzz.
Yeah.
Tell us everything about spermidine.
Well, so we studied that a while ago when I was at the Buck, and we wanted to look at. We were confused because at that time the data was spermidine could extend lifespan, but it didn't impact metabolism. And I'm like, almost everything in a mouse that extends lifespan has some impact on metabolism. So we did a high-fat study with spermidine, and we showed that in old animals, spermidine could suppress the metabolic dysfunction that came from high-fat diet in mice. But we had some control mice too, and actually not many, but because the study wasn't designed to do lifespan, but we left them alive and looked at survival, and the spermidine extended the lifespan of the mice too.
Has spermidine been studied in the ITP? I don't think it has it?
Don't think it has. Don't quote me, but I don't think it has. And so we were able to repeat the lifespan effect in mice, even though that wasn't the goal of our study, and show that it restores metabolism a calorie challenged context. And so that's what made me believe that it's a robust molecule that we can see that effect as well. So I would say I'm optimistic about spermidine as well. That we haven't done a lot.
Where does spermidine occur in nature?
I actually don't remember the answer.
But it's naturally occurring.
It does naturally occur. Yes.
So there's no.
I think it's in certain foods or have it, but I'm worried I'm conflating that with the urolithin. So
okay. Have you guys looked at alpha estradiol 17?
No, we haven't looked at that. I
that
wish
would
we
be an
looked at
interesting
it.
one to look at.
I agree. And I'm also director of an Asian Center for Reproductive Longevity and Equality in U.S. and Singapore, where we're looking at ovarian aging. Mm-hmm. And there's two things I've learned. It's made me, this is like, it's like PhD biologist, all of a sudden I'm getting asked a million questions about HRT. So I decided I better learn something. But there's two things that I've learned from this. One is that geroprotectors tend to extend fertility in mice. And It's not just one thing. It's spermidine, it's AKG. It's been shown for metformin and rapamycin. And
there's a RAPA trial going on, I think, at Columbia or Cornell.
So, And so that's one really potentially useful avenue of translation for these geroprotectors. I'm really excited about that. The other thing I think, and maybe you have more expertise than I do, so please disagree, is that the question should not be, should a woman do HRT? The question should be, is there any reason a woman should not do HRT? Because I just see the value of hormone replacement to far outweigh the risk in the majority of women. I don't know if you feel that way, but it's something
I do.
And so 17 alpha is interesting, right? Because it worked in males.
And not females.
It's also non-feminizing. I don't know what that really means, but I'm not sure how much I believe that, but and so it could be triggering some of these same pathways. And I know, testosterone is another one that, the NIA did the study on it and showed it's a little bit of an increased risk in prostate cancer. And then everybody said, don't do testosterone replacement. And I think that
although that's been fully debunked
I agree.
Now.
Then so most men are not doing that, or if they're doing it in uncontrolled ways and taking levels maybe to too high that could be dangerous. And so I feel like that there's I feel like
men are in a better position today because, one, more trials have been done to undo the bad ones. So the Traverse trial, which was published, it's probably been a year now, but that sort of undid a lot of even though it was the Traverse had many problems with it, but it undid a lot of the damage of some of the fear-mongering from really bad studies that suggested testosterone was causing prostate cancer. Turns out it's the exact opposite. But women, unfortunately, are still struggling under the dark cloud of the Women's Health Initiative, which was, apocryphal. And it just, no one's undone it.
And I travel to 25 countries a year, and a lot of countries in Southeast Asia. And so now everywhere I go, if I see a doctor, I'm like, how many women are doing HRT?
And what are you finding?
Extremely low, like extremely
and because
low.
They're deferential to U.S. recommendations, or why do you?
I think the changes and the what doctors tell people is slower in these countries, but maybe there's less risk. It's also costs money, right? So if you have somebody that doesn't have a lot of access to finances, they may be less able to do it in these countries. I don't know all the reasons, but I'm just looking into it now. But it's stunning how low the HRT is around the world still, and we're just missing an easy opportunity there, I think, to help people.
I agree. I think there's a part of me that is just so interested in the frontier and like, how do you push the boundaries of this stuff? I truly could be infinitely happy working nowhere but at the frontiers of thinking about the molecules. But at the same time, I feel just as excited about trying to figure out how to make sure people aren't scoring own goals all day. Yeah. Right? Like there's so many ways to just help a person without anything magical find another 5 years of life and dramatically improve health span through judicious use of everything from HRT to correct exercise, reasonable nutrition. Like there's just so many great ways to do it. But obviously the idea of doing both of these is so appealing.
I come back every 3 months to the U.S. I lived here 50 years and then I left. And I come back every 3 months. And the same two things, you leave someplace, you come back, you notice things you didn't notice, right? And the same two things every time I come back, I get it in the airport. I don't even have to leave the airport. One is so many people are not in health, they're not in shape. There's so much obesity. It's striking compared to almost anywhere else in the world. The second one is people just seem so stressed here. I don't get that. Singapore is a pretty stressed country, and I still feel that people are more stressed here when I come back.
What are the obesity rates in Singapore?
Relatively low, but in Asia, you've got this challenge with skinny diabetes, right?
Yep.
And so you have a lot of people that can't build the adiposity that they need.
Right, but they're storing all the visceral fat.
So they're storing the fat in the wrong places, and it's maybe even worse than. So the diet is moving more Western in Asia, and it's creating problems. It's just not as obesity associated. So.
And now that you're coming back, do you notice a difference at all based on SGLT2 inhibitors? GLP-1 agonists?
I think they're making a difference, but I guess, I'm just
It's not passing the airport test yet?
It's not passing the airport test yet, yeah. I don't know how widespread they're being used either. You probably know that.
I don't think I'm familiar with the latest stats on how widely they're being used. Let's go back to the epigenetic clock situation. So do you think that there could be value in this as a tool, right? And let me hold the bar as high as I think it would need to be to justify their use. So right now we have this thing called chronologic age. I can look at your birth certificate. I can know how old you are. And based on that, I can make an estimate of how much longer you will live. Mm-hmm. So if I look at a person who's 40 years old and I know nothing else about them, and then I see another person who is 65 years old and I know nothing else about them, I can say with a high degree of confidence that the 65-year-old person will live, I'm making this up because I'm not an actuary, but somewhere between 20 and 30 more years. Yeah. And the 40-year-old person, I can say with a pretty high degree of confidence, will live, somewhere between, call it, 30 to 50 more years or something like that. Now, to me, that's a pretty good test, right? I know a knowable, measurable thing about them. It's measurable by knowing their birth date, right? And it predicts future life. Mm-hmm. So do you think biologic clocks will ever serve a purpose like that, where I could take two 50-year-olds and one of them has a biologic age of 40 and one of them has a biologic age of 60 according to the clock, and that those numbers will actually be better at predicting future life than their chronologic age of 50? Or do you put yourself in the camp that says, no, Peter, that's a ridiculous standard that no biologic clock could ever come to, but it might tell me about their health. It might be yet another biomarker that says, hey, the guy at 40 is just healthier than the guy at 60. And somehow, by the way, it's picking that signal out of a data field that I can't pick out anywhere else because they otherwise look identical.
We measured this recently because collaborators of mine at NUS, Jan Gruber and Feng Sheng, and I had this small role in this project. They decided, Feng Sheng's a geriatrician. He sees people all the time, frustrated. The geriatricians have limited things they can do. They're seeing people that already have multimorbidity, and the clocks are not that useful. And so he wanted to how do we create a reliable clock that a doctor can understand? And so for what purpose? For biologic age. I'll tell you what I think the purpose is for it in a minute.
Okay.
And this so they're first generation and second generation clocks. The first generation clocks try to predict your chronologic age, and the second ones predict some outcome. So the question is, we want to predict mortality. We don't want to predict your chronologic age.
Yep.
So intrinsically, if it works, it's going to do better than chronologic age for the second generation clocks. So we took NHANES, data collected around 1999, 2000, mortality data for 200 months. And we just and these parameters are nice, and because you can actually do a consumer test of HbA1c.
Yep.
There are many labs that do that. It's reproducible to a large extent, much better than DNA methylation. And doctors use all these parameters. So the things that are in NHANES, like LDL, all the things in your book, inflammatory markers, medical tests, some cognitive self-reported stuff. So we took everything as a feature and used AI, linear model, to try to predict mortality. We're on the second generation of this clock now. And it predicts mortality better than any other parameter in NHANES. It's way better than ASCVD, there's cardiovascular disease measurement. And so recently the methylation data came out on NHANES. And so we could go back and compare the mortality prediction for methylation clocks. Some of the first generation clocks are worse than chronologic age. Your passport is better than they are at predicting mortality, which to me means that they're not useful because, even if they're not designed to predict mortality, if they don't capture some element of that, what are they measuring? The second generation clocks like Grim age and Pheno age, they do better job than chronologic age for sure.
At predicting mortality?
Predicting mortality, yeah.
And just to be clear, we've captured that out of the NHANES database?
That's from NHANES. That's the only thing we've looked at right now.
Okay, let me make sure I understand that. You had 200 months of forward-looking data.
Yeah.
So anyway, you've got 18 years of data, and you're saying if we know the methylation of somebody at that time in the cohort, 1999 to 2000, we could predict their date of death better than the actuarial data of their age.
Yeah.
Okay, I wasn't aware of that.
Well, I don't think it's been accepted for publication.
Okay,
I'm not even sure it's online yet. So,
okay.
It's not your fault.
That's very interesting. Okay, yeah. So that basically answers a question that I've never seen answered.
Yeah, but our clinical chemistry clock does better than those.
Okay.
And
what is included in that clinical clock?
There are about 50 parameters that we measure now, but complete blood count gives you about 30 of those parameters, so it's not as elaborate as you think it would be.
Yeah. No, I believe it.
And then, it's a lot of standard markers that you already measure. So you probably measure all of them in your
guess that was going to be my question.
Yeah. It's about $300 in Singapore if you did it all de novo, but anybody going to a doctor's office has most of those parameters measured anyway. And if they're going to, a wellness longevity center, all of them are probably
Of course, yeah. And so the question, I suppose, is this. If you're MetLife, you already, you are better at predicting mortality than anybody on the planet. And I don't know if it's MetLife, by the way, but pick the best life insurance company.
Yeah.
This is their business.
Yeah.
They're so good at predicting mortality. It's frightening.
Yeah.
And so...
Yeah, I don't know how they're doing that, so I can't comment directly.
And nobody does, right?
Yeah.
But my point is they're looking at age.
Yeah.
They're looking at a whole bunch of things in your medical history.
Yeah.
They're looking at a whole bunch of blood tests, your blood pressure, your weight, your waist, your circumference, all those things. And they're coming up with an exceptional prediction of remaining years on life. The real question is, do you believe that they will incorporate a second generation epigenetic clock, or do you believe that they've already got that captured in their data set?
They may, I don't know. It's an unanswerable question. But again, we're focusing on the clinical chemistry anyway. We're not doing any methylation.
Yep.
So, and what we're finding is hospitals want to use this now.
Clinical chemistry or methylation?
The clinical chemistry. And I think it's because it resonates. When you show them the list of parameters, a doctor doesn't have to be an expert in epigenetics to figure out what's going on. And the other thing is they're all actionable. So we have principal components that we can break it down in, and we can see smoking in one component, and we can see metabolic disease in another one, and obesity in another one. And we find cases. So a few conclusions from this are really interesting. One is we find cases where nothing's out of the reference range, okay? So a doctor that's looking at things, especially if they have a few minutes to look at, they're not going to prescribe anything for this person.
Yeah.
But these 4 parameters in this principal component are increasing, their biologic age by 4 years, which means it's increasing their, it's 50% increase in mortality risk. So these are actionable things. You can treat LDL, you can treat high blood pressure, you can treat these markers, right? And so clinicians are actually willing to then be a little bit more aggressive and try to prescribe something or lifestyle modification or something to treat these markers. So yeah, it's actionable.
Yeah, exactly. That makes sense. The reason that you prefer this is it doesn't just give you an answer, it gives you a solution.
Yeah, that's what we're working toward. And so the other thing we did is we broke NHANES down, really interesting. So NHANES also has all the medications people are taking. But the weakness of it is a snapshot. It's a cross-sectional measurement of all these things, but they measured a lot of stuff. And so we could look at what are people's clinical parameters out of the reference range. Should they be being prescribed some drug and they're not being given it? And that goes up to about 20% and when you're 65 years old in year 2000 in the U.S., meaning 25% of people should be treated for something, but they're not being treated. Those people have a higher biologic age and faster mortality, not surprising. There's also the group that has everything, at 65, every clinical parameter looks good. And they have a lower biologic age, they live longer. But you can break that group down and you can say one group is not taking any medication and the other group is taking medication. It's just their clinical parameters are managed well. The people taking the medication have a lower biologic age and live longer. And it doesn't really matter what the medication is. It's true for the major medications you would give for metabolic and cardiovascular disease in the year 2000. So I think that suggests that being more aggressive and getting people optimized earlier is better than just being, I'm pretty healthy and my blood pressure is a little bit high, but I don't need to take, and so it's suggesting we need to be more aggressive. I think the other thing is that it's suggesting that these drugs that were around in 2000 for these treating preconditions are actually aging drugs. They're actually extending lifespan.
And what are the classes that we see the most common? Lipid and hypertension?
Hypertension, metformin, and it's the standard things. And so
do you think metformin will have geroprotective properties in people who are metabolically healthy?
Skeptical. But I think what it is saying is that if you catch preconditions early enough, you protect against the other failure states a little bit too. So it's
So you're more optimistic that rapamycin would be geroprotective in humans than metformin?
Yeah, that would be my prediction, yeah.
Do you think there is a drug out there that you think is more likely to be geroprotective in humans than rapamycin at this time?
Well, the GLP drugs and SGLT2, I think, are interesting. I don't think we have the data right now, but it's definitely, again, I don't know in people that are, if you're obese.
Okay. So let me restate the question. If we just took a population of middle-aged, healthy individuals, and we could design an experiment where you had the placebo arm, the metformin arm, the rapamycin arm, the SGLT2 inhibitor arm, and the GLP-1 agonist arm. What is your prediction in length of life or additional years of life given in that 6-arm study or whatever it is?
I think the last three would be comparable. Metformin, I'm skeptical of.
So Rapa, SGLT2, GLP-1.
Yeah, we don't have data in healthy people that much with SGLT2 and GLP-1. I mean,
so mechanism of action is what? If the GLP-1 group and the SGLT2 group are metabolically healthy, they don't have glucose excursions that are high. They're completely insulin sensitive. What do you believe is the mechanism of action?
I guess I guess I'm going by a different statement, which is most people we're calling healthy are not totally metabolically healthy.
Fair.
And so in those cases, I think there would be a benefit.
But then I guess I don't know
in the perfectly optimized person whether there'd be a benefit. And I every time I talk to a doctor, I ask them, "Are you losing more lean muscle mass with these drugs than you are just by fasting or lifestyle?" And if I ask 10 doctors, I get 10 answers. So I don't know what the answer to that question is. So.
But I think the reason that, at least as a thought experiment, it's an interesting question is because if the GLP-1 agonists and SGLT2 inhibitors only work if you have some degree of glucose irregularity, then it begs the question. Well, it says, look, glucose homeostasis is one of the most important features of living. Great. But if you could correct that with diet, sleep, and exercise, which you can.
Yeah, I agree. You can.
It's hard, but it could be done.
Yeah.
Then those things aren't going after fundamental pillars of aging, because people who eat well, who exercise well, and sleep well still age.
Yeah, it's answering, Matt and I debate this all the time about, and we published a study in mice recently where we analyzed all the data that's out there in mice. And we tried to determine, where's reality on interventions that extend lifespan in mice? Because if the control mice are really short-lived, which happens a lot.
Why is that?
It's just really hard to control. I mean, if you look at the ITP data, the control mice are all over the board, and they're very well controlled. Yep. The best scientists doing the experiment. We see a lot of variation too. I just think there are some cases they're bad vivariums, and that causes a problem. But even in good vivariums, I don't know. It's true in every organism, in yeast and worms. One cohort of worms will all live a little bit shorter. One cohort of worms will live a little bit longer. It's cohort dependent, but I don't know why. But anyway, if the mice are short-lived, if your extension is there, all you can say is it's longevity normalizing. You don't know that it's slowing aging. It's only when the controls are really long-lived and you're getting extension that you can really make the argument it's longevity extending. And so that gets to your question. The real answer, though, is dependent on how many people you believe that are optimized right now, because it's impossible to do the study.
Yeah,
right?
no, of course. It
longevity
is
normalizing works in this population. Yeah. Trust me, at least for keeping people healthy. But whether it's really slowing aging is a open question. I think the best case would be rapamycin there.
Yeah, and then of course it begs the question, which is, could these effects be additive? Right.
Yeah.
So would there be a benefit to a person who is on balance quite healthy, but let's say their hemoglobin A1C, if it is indeed an accurate representation of their average glucose, is 5.4%. Yeah. So I don't know exactly what that translates to. It probably translates to an average blood glucose of 110 or so milligrams per deciliter. Yeah. But the data, there are data that show, based on hemoglobin A1C, that lower is always better. Yep. So 5.0 is better than 5.4, even though 5.4 is deemed completely healthy. Yep. And that's all-cause mortality data. So we're saying we take a person who's at 5.4. They're not even pre-diabetic. They can barely see where pre-diabetic starts, let alone diabetic. But we give them an SGLT2 inhibitor. I'm
at 5.4. I can volunteer for this.
There you go. So you go from 5.4 down to 5.1 just on the basis of that drug. Yeah. We throw a GLP-1 agonist on top of that. Yeah. Now you're at 4.9. Yeah. Then we give you rapamycin, which really doesn't impact your glucose, but we think it's going to do something a little bit different. You would say in that situation, you might believe that there's some actual
I think it's
geroprotection,
feasible.
but not adding metformin at
I
that
don't want to
point.
lose muscle mass, though.
And which of those drugs would you be most afraid of, the GLP-1 agonist or Rapa?
GLP-1 agonist.
Okay.
I think lean muscle mass is like super important. Yeah, it's probably better to have high lean muscle mass and be a little bit more fat than it is to be low on both, is my best guess.
Now, what do you make of the data that I talk about all the time, which look at the hazard ratios for mortality based on high VO2 max and high muscle mass and high strength, and how those three things stand out so far above anything else? Meaning, like when you look at hazard ratios associated with smoking, type 2 diabetes, even cancer, they are not as lethal as being incredibly weak, incredibly low in muscle mass, and incredibly low in fitness. Yeah. And how much causality do you think is there versus how much of that is just, those are just such good markers of health?
I think there's causality there. I think it's super important, but it may only be important for squaring the curve. I don't think there's much evidence that maximum lifespan is extended by these things. And we don't have the human data, of course. The animal data.
We can't do the experiment.
The animal data pretty much with exercise says you square the curve. Well, again, I agree with you. It's a revolution if we can do that. I'm not being negative about it, but I don't know about maximum lifespan, whether there be an effect or not. But I believe it so much that I put a lot of effort in increasing my lean mass, and so that's why I started resistance training, because I wasn't getting as much from running. I get more mindfulness from running.
Yeah.
I get, but I did a lot more resistance training too. So.
Yeah, I typically will tell patients that you should really think of exercise. Yeah, I do actually think it's reducing your risk of chronic disease, but then you still get into the whack-a-mole game. If it lowers your risk of Alzheimer's disease, it might not have much of an impact on cancer risk. It's unclear. But if it lowered your life expectancy by 6 months, it would still be worth it based on the health span benefits that you get and the quality of life that you would enjoy, especially in that final decade of life.
Probably. Let me come back to one point, though, because I'm a bit of a rant about this. Combining interventions. First of all, I will say two things before I say what I'm going to say. One is that I believe we need to empower people to make decisions on their own health. And so I support hackers. If they want to educate themselves and try different things and they know what the benefits and risks might be and what we know and we don't know, more power to them. I feel like part of the reasons we get such low compliance in medications is that we don't empower people. We don't give them choices. They don't know why they're doing things. We just tell them what to do and people don't respond well to that. So having said that, I can't pick 3 interventions that work well together in a mouse, and we do these studies all the time. They're more likely to cancel each other out than to have additive effects. And so if you're mixing 20 colors or taking 20 pills, it's like mixing 20 colors of paint together. You're going to get some ugly gray outcome, or at best you're going to get an unknown outcome that we can't predict. So I'm really cautious and want to tell people that there are a lot of people out there promoting doing a million different things at the same time. I try 1 or 2 things at the same time. And I try to see how my body responds. I measure things, even simple measures are useful. And I think that if you're doing 10 things, you don't have any idea what's working and what's not working and whether things might be impairing each other. And I really think that's a scary path to go down.
Now, what about, for example, how are you deciding to use the alpha-ketoglutarate and the NAD? Because you've seen at least some evidence that each of those individually works.
Yeah, I've been using the Rejuvenate with the AKG for a long time because I was involved in the research. I'm actually on the board of the company. It's something I've done and I've just taken for years. And I add one thing to it and take it away. So I've tried astaxanthin, I've tried fucoidin, I've tried rapamycin, I've tried a bunch of other things too. So, and I try to measure before and after. I'm getting better at that. When I first started doing it, I wasn't measuring that much, but and so that's the approach I take. But I never take like 6 things at one time. And it's kind of intuitive. If something's interesting to me, you see an effect on the mice, I want to try it. So like I want to do urolithin and astaxanthin next. And
if you had significantly more resources, right? So let's say you had a budget that is, what is your current budget this year for both animal and human research?
It's complicated because we have multiple streams. I would say we probably spend about $4 million a year total.
Okay. So if that number were multiplied by 10 or 20, like if you had 25, you had $100 million annual budget to do world-changing translational geroscience, what would you be doing different?
I think the scale would be one. We just can't. When you do a combination in mice, you've got 4 groups. And I think if we had more money, we could design sort of multifactorial clinical studies and preclinical studies where we're testing many compounds at the same time. We're sort of doing nested groups, and we could get an indication for things that actually could be additive together, not just things that are working on their own. Right now, we struggle to get to that next step for finances, and we could apply those to human studies and combine it with lifestyle interventions too. The other thing we really believe is that when you have a compound like urolithin, you're never going to really know what to combine it with unless you know what the compound's doing. So we do a lot of discovery stuff now, trying to figure out not what pathway. If you take a drug like rapamycin, it affects every hallmark. That doesn't tell you the primary thing the drug is doing. In this case, we know it binds TOR. Urolithin, we don't know. So we need to know what that molecule is binding to in the cell. And if we understand the mechanism at that level, we can combine it better with other interventions and start to understand how to put the puzzle together of what we need to combine to get the biggest effect.
Do you have enough human resources to deploy that kind of capital if it were available?
It would take a center to do it, but yeah, we could build it. And Singapore is very motivated, by the way. I have to give the government credit. They've understood the aging problem before almost anybody. It's taken them a long time to really figure out what to do about it. And, so they went through kind of early stages of putting roofs on sidewalks so people walk an extra 100 steps in the hot sun. That sort of stuff helps. But, but now I think they're really motivated to commit to targeting healthspan, which if they do, it's the right place to be because it's a small island. It's a compliant population. They believe in their government.
What's the population?
It's 5 million, and then 1 million workers and 5 million people that live there permanently. I think that it's a good place to really take studies, not just in the clinic, but move them into the community and actually get validation in large populations. And that's why I like being in Singapore. I think the opportunity's there to make it an example for how to do healthspan. It's already very long-lived, by the way. So, it's among the top 3 in the world, depending on what statistics you want. But the healthspan, the people still have morbidity there. They still have 10, 12 years of sickness and decline. There's a lot of frailty there. You can see that just walking around. So there's room for improvement for sure.
How will AI help in this field? Do you think it will allow for more intelligent experiments? Will it allow for better signal detection in messy data? How do we unleash AI on this problem?
We're using it now already for clocks and signal detection. We're using it to pick drugs now. Where we just published a paper with Georg Fullen in Germany where we're trying to improve how to ask large language models medical questions related to longevity. I talk to a lot of doctors and they say you shouldn't be asking perplexity these questions. And I'm like, I'm a realist. People are asking. So figure out how to get them the questions asked in the right way to get the right answer.
But I'm thinking more in terms of the research.
No, I'm just giving you examples there. I think that what's going to happen next is AI is going to start telling us what questions to ask. Right now, it's telling us how to analyze our data.
Yep.
It's still not very good at telling us what the next question is, and I think that's the sort of the threshold.
How do we get it there? This is a very difficult question.
It's not one that I'm qualified to answer, but I think that the question is the trajectory that it's on now is amazing. Is there a barrier to go to that next step, or is it just a matter of getting computational power and slightly modifying neural network algorithms or something? And I don't know the answer to that, but I do think there's a reasonable chance that I'm not going to be needed in 10 years. So.
I think about this question a lot when it comes to experimental topics because I still don't have a good enough sense of how many training cases an AI needs to learn this. Like we know for language what it took.
Yeah.
We understand how many tokens were needed to allow the neural networks to do what they do today, and it was enormous.
Yeah.
So there are some problems that might require far less input.
Yeah.
You might be able to do it with 10,000 hours of data.
Yeah.
As opposed to billions of hours of data.
Yeah.
So to me, I think that's the question more than anything else.
Yeah.
I was wondering if you had a point of view on the answer.
Yeah, I don't really know. I all I know is half my lab is doing it now. So it's I never would have guessed that 5 years ago.
Meaning they're trying to use AI to help them ask experimental questions.
Yeah, or interpret data.
Okay. The latter I can understand.
We've become half dry lab, which I never would have guessed in my lab. So it's meaning that half the people just sit at computers and aren't doing experiments in animals.
Yeah. So what are you most excited about trying to uncover truth or high probability of truth in the next 5 years?
I think experimentally, at least preclinically, we want to find interventions that really combine together to have synergistic impacts. I don't think there's much out there. There's rapamycin and metformin and a couple other things from the ITP, but they're small effects. Can we break through a barrier and get 50, 60% effects in mice by combining things together? I think that's what preclinically we're excited about. And the other thing I'm excited about is going back to this entropy question. Are there new classes of interventions that change that primary linear accumulation of quote unquote damage? I'm also really excited now that I'm working with longevity clinics in various countries. And so we're applying the clock we built. We're also helping them try to decide which interventions to do and hopefully collect data so it can be analyzed. Because I think there's so much going on. I tell this joke all the time. I'm getting scared to tell it more, but it used to be that yeast and worms and flies were the model organisms for aging research, and now billionaires are the model organisms because they're doing all kinds of stuff I can't even test. And I'm really curious to see what's happening. Some of it I guess might work, some of it I guess might not work, but we can't find out any other way. And I don't want to see these clinics working in isolation and nobody's ever at least learning what's coming out of the data, even if it's not perfect. So I'm excited to work with these clinics and go see what they're doing right now. That's maybe not the answer to your question, but there's a lot of cutting edge stuff going on. And I will pretty much work with people if they're doing something I consider safe and if they're honest about the data on efficacy. Those are the two things I ask: transparency and safety. And then I'm happy to try to interact.
What are you seeing that you're worried about? What trends do you see that people are doing from a biohacking longevity standpoint that have you concerned? And I'll put this in two buckets. The higher bucket would be safety. Yeah. The second bucket would be predatory behavior around basically people getting having their money wasted, even if the agents that are being sold are not necessarily harmful.
Yeah, I think the first one, I'm excited about gene therapy. Don't get me wrong. I think it's interesting and it may really change the field going forward. I even kind of like follistatin,
Follistatin as a protein.
Yeah. But I think that those treatments are not very well proven yet, and I would not do it. I asked the question, would I do it myself?
You wouldn't spend $100,000 for follistatin gene therapy?
I probably don't have to spend the money, and I still haven't done it. So, I've done MSCs though, IV to try that, and I think stem cells. It's a different question there. I think if you're repairing soft tissue damage or something like that and injecting them directly, it probably works. For aging, I have no idea, but I think it's probably safe if you have somebody that's a good practitioner that knows what they're doing. But I think there's this is a problem with stem cells. You go places and you really don't know who you're working with and, if they're really treating the cells correctly, if you're putting the right things in your system. And so there's a safety concern there based on the practitioner, I think. And so those are things that concern me. I haven't gotten totally on board with growth hormone yet. I mean, I think probably used correctly might be okay. The data is interesting.
Are you aware of human data that, and I'm in your camp, by the way, which is, I actually had a bunch of friends over for dinner last night and this came up and I was sort of, I said, look, I can't point to a study that tells you this is a bad idea. Yeah. And I've never spoken to a person who takes a modest, judicious dose of growth hormone who doesn't tell me they feel better.
Yeah, I agree.
So it's hard to believe it's not making people feel better. Yeah. I also have never seen data to suggest it initiates cancer.
Yeah.
But it seems very biologically plausible that if you have cancer, small amounts of cancer, your probability that this becomes clinically significant is higher in that camp.
Seems like it. I agree.
Again, I say that with no data.
Yeah, me too.
And so, my view has just been, despite my own interest in trying things, I've left the growth hormone one off the list. Mm-hmm. Is that an answerable question, do you think?
Yeah, and I think we could do clinical studies on this, and some are being done.
How would we address
small.
the safety concern, right? Because you really need to be able to try to track people for quite a long period of time.
That's the problem with the gene
cancer
therapy
susceptible.
And all of these things, right? We don't know what the long term is, and I guess it's a gray area. This is a weird thing with these clinics, right? Because there's, I think, from what I read in your book, you're doing sort of validated stuff. You're not really out there in the stratosphere doing crazy stuff that we don't know about. Some of that
I would like to think I'm not, but I'll tell you, there are people who are very critical of my use of rapamycin in patients for geroprotective reasons. There are people who might think I'm crazy for giving people SGLT2 inhibitors who don't have diabetes. So I think there's things that we do, not for all of our patients. And I fewer than 10% of our patients take rapamycin because my view is unless you're willing to have a very lengthy discussion about the pros, the cons, the risks, the uncertainties, and I don't give people an answer that says, oh, this stuff's amazing. My answer is, I don't know. Here's how I think about it probabilistically. Here are the trade-offs. And again, after you can tell I'm not a good salesman if only 10% of the patients are taking it.
Look, I think that's perfectly reasonable. But there are clinics doing some really out there stuff, how do we know the long-term safety on it? We just, I think if you're going to go do that stuff, you need to go in with your eyes open. You're taking a risk.
I've just seen so many horror stories of people that have come back from parts of because you can't do this. A lot of the stuff you can't even do in the United States. So they're coming back from South America or Mexico, places in Asia, having done follistatin therapy or other sort of very questionable stem cell therapies. And I mean, people that have had horrible infections, like literally just artifacts of the treatment.
Yeah,
never
again,
mind.
That's a practitioner problem with this.
Yeah, exactly.
But there's some great stuff happening too. I work with Bumrungrad Hospital in Thailand, and they've got a longevity clinic now, and they're very grounded in good science. And so, but the problem is if you're a consumer for these products, it's really hard to know.
So how someone listening to us who's saying, guys, can you give me some rules of thumb, some heuristics for navigating the never-ending landscape of longevity hacks that keep showing up on my Instagram feed, my TikTok feed, and at cocktail parties.
Yeah, I mean that could be diet books, not just going to clinics around the world. And I think it's really difficult to sort that out, and because what I was going to say is it's clinical practice and research at the same time. It's a very unique situation, right? And there aren't many examples of that I know of that are really, maybe some functional medicine is a little bit like that too, but it's interesting, and I feel like it's better for scientists to engage with, where it's possible to engage with these clinics and try to help them than it is to just let people do things and without, if you can provide oversight that's helpful, you should be doing it. And so that's kind of how I feel about it. But it's a lot of academics don't even want to work with these clinics at all too. So so I get criticized for working with them sometimes. So it's an interesting world right now.
Well, there are a lot of things we talked about today, Brian, that I can't wait to follow up on myself. So I'm really looking forward to the NHANES second gen clock paper. That'll be interesting. Again, my personal curiosity there will be, is that clock providing value over all the other data we have? My intuition is it won't, but the fact that we now at least have a clock that can outperform biologic age, chronologic age is a step in the right direction. I'm going to be very interesting to see some of the data that you've talked about as far as alpha-ketoglutarate. You've also piqued my curiosity with the sublingual NAD. I'm sorry, and so that's really interesting. Have you been able to measure NAD levels in your RBCs? And
I haven't done
that?
it myself. There have been some studies done
on that exact supplement.
Yes, I don't think anything is published. It's done by the company, but
and I do like this idea of taking ITP winners and combining them and seeing if we can get, if you could get accretive value, that would be remarkable. But even if you could just get additive benefit, it would be pretty amazing.
Yeah, I think so too right now. I think that's still a hang up for the field.
Well, this was super interesting, Brian. I really appreciate you making the time to come out here.
Oh.
I know that being on a plane for that long is not always fun, but
that's where I live anyway, so it's fine.
Thank you.
Thanks a lot.